This week on Surfing the Nash Tsunami.
The core of Nash Tag is a vigorous exchange of ideas. So if you have a question, if you have some heretical perspective, send it in. We'll be looking through and get to as many as we can, but speak up and agitate. The field needs to move forward. It needs change.
While the surfers prepare for this weekend's Nash Tag conference, Roger Green interviews Nash Tag co-directors Vlad Ratziu and Michael Charlton about the history of Nash Tag and their goals and hopes for this year's meeting. Roger then discusses how SurfingNASH plans to cover Nash Tag, all coming up this week on the Surfing the Nash Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 2, Episode 10 of Surfing the Nash Tsunami starts now. Hi, this is Roger Green, executive producer and host of the Surfing the Nash Tsunami podcast. This week's episode will be shorter and a little different than what you're accustomed to these days. We figure that with 1 hour of Nash Tag programming on Friday, 2 more episodes on Saturday, 1 on Sunday, and then a wrap-up next Wednesday night, you'll have all the programming and conversation you need to listen to over the next few days. So instead of doing a full episode tonight that would produce a lot of conversations nobody had time to hear, we decided to, as Stephen talks about sometimes, apply the KISS rule, keep it simple, stupid. Stephen, who's one of the Nash Tag co-directors, walked us through the meeting on episode 8 a week and a half ago. Tonight, we're going to have interviews with 2 of the other co-directors, Vlad Ratziu and Michael Charlton, who will talk about how they got involved with the program and what makes them excited about what's going to be going on this next week in Park City. After that, I will come back and spend 3 or 5 minutes letting you know about the specifics of our coverage for the meeting. So with that, let me be brief, and I will turn to my interview with our good friend Vlad Ratziu. See you later in this episode.
And now, a conversation with Nash Tag co-director Vlad Ratziu.
I'm here with Vlad Ratziu, who is one of the co-directors of Nash Tag, and I believe has been since it started, right, Vlad?
Yes. This is now at least a 5-year-old story. Started more than 5 years ago, actually. And I'm one of the initial co-directors.
I commented to Vlad that I'm fairly confident that I am not the most disappointed person not to be there in person, because I know that Vlad is even more disappointed than I am that he cannot get over from Paris for this. But and we may touch on that as we chat. Tell us a little bit about how Nash Tag formed and how you came to be involved with it in the first place.
So, in retrospect, it's a nice and hopefully successful story. Actually, I went a few times to HepDart, which was a single topic sort of meeting on viral hepatitis that was held every 2 years in Hawaii. And I liked the model, which was that a lot of people from different horizons were attending the meeting, all trying to understand where we stand at that time with drug development in viral hepatitis, but also help to describe viral hepatitis disease and all these other clinical aspects. And I said, why don't we create such a meeting for NASH? Because NASH already started emerging a little bit less than 10 years ago as a major chronic liver disease. And I found that the time was ripe to have a meeting where different interests in terms of drug development would converge, both on the academic side, on the industry side, where we could have a meeting discussing aspects of the clinical side of the disease, but also aspects of how you diagnose the disease, what kind of drugs you develop, what are the new drugs in development, where people would exchange in a very relaxed setting where there's a lot of some sort of other attractions than the meeting itself, which would allow them to have which would facilitate the exchanges, would facilitate personal interactions, and even build personal friendships. And I thought that was an ideal model to bring people together in a very nice atmosphere, but also at the same time a highly selective place with a very high level of discussions on NASH therapeutics. And that's how it all started. And of course, since I was not able to do it myself alone, we set up a meeting with some of my close friends or collaborators in the United States. So at that time, it was Mike Charlton, Stephen Harrison, Arun Sanyal, and they all found that this idea would be interesting and we should give it a try. Of course, there were some issues with sponsorship sponsorship in the very beginning. But those were worked out. Everybody was very enthusiastic, also friends from the industry. And that's how we set up the first Nash Tag meeting. We decided on a model where we would have the meeting in winter, where people could enjoy the winter sports, including skiing. So we had to find a location in the mountains, and it never changed since. We still hold it in Park City, in Utah, in the first week or the first 2 weeks of January. So it's been a model that people embraced and progressively liked and adhered to. And I think everybody has something that they like in this meeting, and they take home, in particular, the very nice, friendly environment that brings these people together.
It's pretty unique. And, I did marketing research for most of my career, so I've seen just about every major disease class and kind of meeting that exists. And in the first few years before the PD-1s became big, SITC was this kind of meeting. The Immunotherapeutics and Cancer meeting had this kind of feel to it. But then all those drugs exploded. Maybe it's because the drugs haven't exploded yet, although hopefully you can maintain this feeling even then, because it's a pretty unique meeting in a great place.
We're very confident we can maintain it. One of the things we're really striving to keep is the original format. We're trying to make sure that a large part of the conference is being conducted in an academic style. So it's not a commercial conference. So we give a voice and a large part of the presentations and the interactions to academic people. But we also need people from the industry, because in the end, those people working in R&D are the ones who discover the new molecules. Sometimes they discover even the targets themselves when it's not the scientists, the biologists. So we need the expertise of these people. So I think the success of Nash Tag at a very high level is the fact that we were able to assign to each particular aspect of the conference the right people who can talk about that. And we will discuss this a little later about which parts are being presented and discussed and developed by the academics, which part are being discussed by the industry, and what are the other topics. And I know you've been in this conference now for at least the last 3 years.
This will be the third.
This would be the third. And the reason you were there is because we wanted to extend the conference to get input from other fields than just rather the clinical drug development field, but also get some additional insights from the regulators, from the societal perspective, and so on. This was the reason why we invited you, and we invited you back, because we were very happy with your contribution to the meeting. So this was a key aspect, giving the right field and topic to the right person, maintaining the academia industry balance, and not falling into a sort of a commercial format for the meeting.
First of all, thanks for the kind words, and thanks for inviting me. It's been a great experience, and that's why we're doing this podcast, is because of things I started to learn going to Nash Tag. Maybe one way to lead into the meeting would be to ask, so there is no commercial section this year, right? The section that I was part of, that Yasmin, and talking about some payer issues, how payer and policy issues might affect drug development. I don't think that section exists this year.
That section does not exist this year. So we have to rotate a little bit the subject, because after all, it's only a 2-day meeting. It's a rather short meeting, which doesn't mean that subject is not interesting, but we will come back to it certainly in the near future. So there is no commercial part to it. Of course, the exhibitors and the sponsors are granted the possibility to have a booth, but that's about it. The program itself is being designed exclusively by the 4 course directors. And the key thing is that we're very happy to have industry participating. So it's not like we're, for very rigid ideological reasons, keeping them out of the program, but we try to ask them to contribute in the field where they have the real expertise. And I think this blend of best knowledge from the academic side, clinical side, and best competence and knowledge and know-how from the industry side, I think this is what makes it unique. So we sincerely try to put the right balance here and achieve this sort of magic mixture where people talk to the best of their competence about the subjects they know best. See, that is the thing. Because everybody has, at this level, very, very strong knowledge on different things. So it would be silly for me to say that I know things about drug discovery and how to test in preclinical models better than people who do it all the time successfully. So that is the philosophy behind that. Give everyone the possibility to talk in his own field of excellence.
If we were going to pinpoint 1 or 2 sections in this meeting as it's designed this year that you're particularly excited about, which would those be?
So listen, here's the thing. If you have followed the programs, which are actually on the website for the past 5 conferences, right? Because we had already, I believe, 4 or 5 editions of it. You've seen that the main sessions do not change. And we try to maintain that particular distribution of topics. So the conference starts with a clinical session where we present the data on the latest trials. And we chose to discuss only trials with histological, only phase 2 trials in humans, so not very early, but rather phase 2 trials in humans. But now there are so many that we sort of extended that a little bit to more early phase trials. So what is really crucial to understand is that this session, which is, by the way, in the format of a sort of controversy. Someone is presenting the data, usually the physician, the PI that was involved in that study, and someone else does some sort of rebuttal. So this is a clinical back and forth type of discussion on the results of the human trials. And this is exclusively done by academics because those are the PIs that have done the study and some KOLs that have the ability to put that in perspective and provide a slightly different take on things. So this is one session I'm looking forward to because not only it gives you a nice presentation of the results of the main trials, but also it gives you a discordant sometimes point of view of someone else who tries to underline the limits or interpret the findings in a different way. So that session, which is session number 1, has not changed a bit in its format since the very beginning, and we try to keep it the same way. So this session on latest clinical trial results is very important. Now, it all starts with something that everybody is looking forward to each year, which is a sort of a year in review, sort of state-of-the-art lecture by Mike Charlton, who does that really wonderfully each year. Very great overview of the important things that have occurred in the field since last year, since the last edition of Nash Tag, and he puts a lot of humor into this as well. And since he's someone who has a lot of experience in the field, is able to pick up the very important things that happened, present them in a very educational way, makes you understand things that you have not understood, and all this with a lot of British humor. So this is what kicks off the meeting, something that everybody is really enjoying and looking forward to. Then you have the session that I just discussed about latest clinical trial results. And then just to mirror that, the last session of Nash Tag is about drugs in early development. So drugs that have not yet hit the human phase trials, but that look very promising because of their mode of action, because of the data that have been observed in preclinical models. So that is the exact opposite of the first session in the sense that it's only being presented by people who actually develop those drugs. So people from the industry, from the research and development department in the industry. So people who know best the targets and the molecules they are developing to hit those targets. So that is a very exciting session, short presentation around less than 20 minutes, should be 15 minutes plus 5 minutes discussion, where each new drug that we consider is of importance, either in terms of what it achieves, but also in terms of how original mode of action it has, is being presented there. State-of-the-art, cutting-edge new drugs and development gives you the opportunity to be really fully keeping abreast with what's new and what will be prominent in the future of NASH therapeutics. And that is done by the people who actually do the work. It is moderated by academics, but it's done by the people who actually discovered those drugs and tested them in animal models. So see, two very different philosophies, but that explore drug development from the two different ends. And I think if you put the two together, you get a great upgrade of what is cooking and what is in development in humans and what has the best perspectives of success for NASH drugs. So when I say NASH drugs, of course, most of the things that are being presented are about treating NASH, but we also try to not to neglect and actually discuss with the same interest antifibrotic drugs as well. So it's NASH or antifibrotic drugs. Now, of course, a lot of these antifibrotic drugs are being developed in patients that have NASH as an indication, but they have the specificity of targeting fibrosis in particular. So those are the two types of drugs we're discussing. So those are the two things in mirror. And then we have other sessions that are of high interest. This year is an exception because we had so much to discuss on the clinical side. We have no pathophysiology session, but we try to keep each year a very strong session on more fundamental aspects in terms of steatohepatitis or fibrogenesis, where we try to bring the best experts that do research and lab work in these aspects to tell us about new pathways of injury, about new molecular targets of interest. So this is one very important session. And then, of course, the rest of the conference is about, and this is particularly true this year, clinical trials in general. How do you perform clinical trials? What are the obstacles? What kind of innovations are out there in order to better conduct these clinical trials? And this year, this is a large part of the program, as you have certainly seen. I will finally end by saying that we also have something new for the past 3 years, is that we introduced these mini debates. Now, the mini debates are very fun because you basically have two guests, and each one of them is presenting either a pro or a con on a specific subject. And they trigger tremendous discussion in the audience because obviously we're choosing subjects that are not settled, where you can see things in a different way, where there are opposing points of view. And since these very carefully chosen speakers are able to very strongly defend one or the other point of view, that creates a very nice interaction also with the public. And we're trying to develop that each year in this mini debate series.
So Vlad, when I think about the moments that have interested me the most, one of them was the mini debate last year, which was kind of the aftermath to Stephen presenting the paper about the kappa scores and the reliability. And so one of the things we talked when we did the preview on the podcast, one of the things that we talked about that we find interesting is that things start and then bubble up again and again. So last year, for example, that whole idea of reader reliability showed up 2 or 3 different times when Stephen presented the paper. And then when you folks got into the debate about one pathologist versus 2 pathologists, it came back up again. And then there was a third time, I don't remember when it came up. And you could see as the meeting went on that people kept thinking differently about the same subject. The thoughts were evolving in the room in real time, which you never see, and I thought was amazing. As we went through the meeting, people were thinking more and more about this reliability question and the kappa scores. And as they talked to each other and they thought about it more, there was really good intellectual ferment, and people wound up with a slightly different place, some of them, than where they started. When we did the preview episode, we thought, for example, the semaglutide might be that kind of thing. Right? I mean, there's a semaglutide paper the first morning. There's talk about diabetes drugs being the end-all and be-all as a debate on the second day. And the idea that people had such tremendous hope for sema, and that in some ways it's turned out quite fine, but the idea that there's no antifibrotic effect is a question people are asking. I wonder whether you think that might be the kind of issue where people's thoughts will take shape. Another one might be PPARs, or another one might be something that's not about drugs, but about how to conduct good trials. Can you see an area where you think there's going to be that kind of ferment or intellectual growth as we go along? There
are many areas. So first of all, of course, as you said, having the presentation, synthetic presentation of the results and someone challenging the interpretation of those results is very enlightening. So that for sure will benefit a lot of people. Then we have a very large discussion about how trials would look like without histology based on biomarkers. What can we achieve with biomarkers? How far are we from replacing histology with biomarkers? So I think the strength of Nash Tag is indeed that it looks into issues that are very important for drug trials and drug development and analyze them under different perspectives, which gives the opportunity to everybody to stop and think about these issues and maybe discover things that you have not seen before at the first glance. So yes, all these issues, what we're trying to do is to dissect the complexity of conducting clinical trials in NASH, to see what needs to be improved and how we should improve those. And for instance, this year we have a discussion about platform trials, which is a new innovative way to perform trials.
Can you explain a little bit about what a platform trial is? I'm not familiar.
It's a very different way from the traditional trials, which means in a nutshell that you're not doing the trials one by one independently, but you are inserting the patient in an environment where different drugs are tested at the same time, which has a lot of advantages. One of the latest examples is the COVID trials, which has been, in particular, the recovery trial has been performed that way. So we will present that. We will show how this has the potential, at least theoretically, to improve the trials, to improve the cost of the trials, the duration, how fast they are being performed, and how well the results are being collected, and what overall advantage that has in drug development for NASH. So see, we're looking at very technical aspects of drug development and conducting trials in NASH, and we're trying to both discuss issues at stake and bring some new perspectives, some innovations into the discussion. So I think there's plenty for people who are interested in drug development of NASH to take home and think about after having attended this meeting.
So first of all, this has been really great and helpful. And, Stephen walked people through the meeting on one level. I think what you've just done is great and kind of helped out on another level. And the next interview they'll hear is with Mike Charlton, which I'm doing a little bit later today. And I'm sure he'll have a third perspective, and that'll be fantastic. Just to let our listeners know, Vlad will be joining us Monday night for the wrap-up interview, although I'll be talking about that in greater detail later on in this podcast. But Vlad, any other words or thoughts you want to leave listeners with, knowing that A, they may listen to this and decide to enroll virtually, or B, that they're going to be attending anyway, but they're not sure how to balance the meeting up against all the distractions that you get when you're at a virtual meeting?
I think this year is hopefully is an exception, and I hope that next year we can resume physical meetings. This type of meeting gains its full potential when it's a physical meeting. I think it satisfies everybody. It's a limited number of people attending, so a rather small audience, which allows everybody to participate when they're in the room. It is a very nice setting. It creates really very nice bonds and interactions between people, whether when they're discussing in the meeting room or when they're discussing on the ski lifts together or in the evening next to the fireplace. So it gains a lot from being a physical. Now, because this cannot be done obviously this year for a lot of people would still have to concentrate on the content of the meeting, which we hope is very rich and will be appreciated by most of the attendees. So we'll have to concentrate on that. Of course, following virtually a meeting, especially with a very large time difference, it's a challenge. It does have some advantages in the sense that you really see the slides on your computer and you're very close to the presentation itself. But there's so many advantages to the traditional way of conducting these medical meetings that I hope we can go back to physical meetings next year. This is a very rich program, and you'll see that some things that we don't think about every day, but which are very important for clinical trials, are being presented here. And if you go through the program, you'll see the special emphasis on artificial intelligence, the special emphasis on some populations that are important populations for the disease, but are being left out of trials. What should we do about it? People that drink alcohol, people that have HIV could be in some of these trials. Can we accommodate them? So it's a very rich program for those who are interested in drug development and in a larger sense in NASH.
Thanks for your time today. We're all excited very, very much about this, although disappointed we won't be in person. I will buy you an after-dinner drink by the fire next January after we've come off the slopes together. All right?
My pleasure.
Thank you, my friend. We will see you Monday night, and enjoy the conference as best you can, and we'll see you then.
Okay. Take care. Have a nice weekend. Bye.
And now, a conversation with Nash Tag co-director Michael Charlton.
We're here now with Michael Charlton, who is another one of the co-directors. As you may recall, this episode is going to be talking with Vlad and talking with Michael. And Stephen talked at length about the podcast a couple of weeks ago. So, Michael, first of all, welcome, and thanks for joining me this evening. People don't know that we're doing this at our dinner hour, so welcome.
Thank you, Roger.
So just 3 or 4 questions. How did you come to get involved with Nash Tag in the first place?
I was at a medical conference with Vlad Ratziu, and we were skiing together in Alta, maybe the highest temple of skiing in the world. To some. Wonderful snow, great location, quality of snow is good. And Vlad and I were riding a lift together, and we were lamenting that there was a meeting that we had both enjoyed called HepDART that was totally devoted to therapeutic development in the hepatitis B and C and even HIV. I think it started with HIV. I'm not sure. I wasn't there in the early days. And we thought that such a meeting was needed for fatty liver disease. And in addition, it should be somewhere that people wanted to be, not in some conference room next to a hotel where it's time out of life that you don't get back. We wanted it to be a place where people would want to interact and that we had some natural gap in the middle of it, where people had the time built into the conference to network, to discuss all those things. So that was the birth of Nash Tag. It was a ski lift ride together with Vlad Ratziu.
Compared to that initial vision that you had 5 years ago, how's it developed?
It's exceeded any reasonable expectation. We thought it might be a one-off conference, then maybe 2. And now it has a life of its own that if all 4 of us, for whatever reason, were not able to take part, it would still run. I think it has become a unique event on the NASH academic landscape, and it brings together the thought leaders in academia, in the pharmaceutical industry, investors, people who are just interested to attend as well. It's really developed its very own brand, so to speak.
Everyone who listens to the podcast knows this, and I think you and I might have talked about it last year briefly in Deer Valley, but I became enchanted in the whole idea of fatty liver disease going to Nash Tag, in part because you folks did such a great job of explaining so many things, and in part because the conference itself is just a different thing. What do you like best about how it's developed?
A couple of things. That's a good question. One of the things I like is the lack of formality. It is a crucible. People will present data, people's data will get presented, and it will be discussed in great detail, analyzed at a level that you don't see anywhere else. There's really no other venue that you can go to or listen to now with this hybrid medium that we have, where you will have this sort of analysis and discussion. Things that might seem almost rude at ASLD or EASL are not rude at Nash Tag. They're friendly, tough questions that everyone has to ask. At some point, people have to ask these questions. And that happens in a way at Nash Tag I don't see anywhere else. And then there's this very friendly backwards and forwards between the directors, between speakers and attendees. You've seen it. It's a very vigorous, lively forum.
Yeah, I agree. One of the things that's struck me, and I've seen it 2 years in a row, is the way ideas really morph over the course of the conference. The best example, I think, being last year when Stephen put up his paper about kappa scores and reader reliability. So he puts it up and people go, And then the next day there's this debate about 1 versus 2 readers in pathology, which turns into in part that, but in part a discussion about why does any of this matter if the kappa scores are so crummy. And by the end of the conference, people are talking about the problems with the previously accepted gold standard. Not that the problems were new, but the skepticism and the dissatisfaction just ramped up day by day. And you felt an urgency at the end of the meeting you never felt at the beginning. I thought that was really unique and a function of what you're talking about.
I agree. And one of the things that we try and do during the years we put together the course is we try and think of the most at the edge of the universe data, whether it's AI interpretation of path, new targets, this kind of thing. And we will bring that to the podium. If it's exciting and potentially paradigm changing, and some things we present are not, but some are. Like we had Path AI from Boston, and I think it's Andrew Beck, the CEO of that company, presenting before they had a single paper out. It's that kind of conference where something that we think can be meaningful, we'll discuss and present at its very earliest form.
I think that's right. The other thing that's unique about it, and Stephen mentioned this in the episode, and I'd be remiss if I didn't, is your opening talk. It's made me 2 years in a row sit up and say, gee, this is different than I thought it was. Not the opening talk, but what you were talking about. Last year, when you talked about the predictiveness of ELF frequently exceeding the predictiveness of biopsy, that was really the first shot across the bow on that entire issue. Stephen was saying in the episode that was a preview that you basically work on it all year.
Oh, that's true. If I hear a great presentation, which I've heard from a presenter, for example, from Imperial College in London, made me think very differently about biomarkers. He was talking about liver cancer, but what he was saying had direct applicability to fatty liver disease, and I'll be presenting some of that in a couple of days.
Will it be on screen with you when you present? I assume he's not coming over. But
no, it was really more of a way of thinking about things. There'll be data presented. And one of the things that has happened in the field in general, and I was here for Hep C, you might have been here during the Hep C therapeutic agent development timeframe as well, where we had advocates, we had patient advocates, banging on desks, interrupting conferences, making people listen to them. So this is important. You have to accelerate the timeline for therapeutic agent development. You cannot have everything developed in series. Sometimes co-formulated drugs are the way to go. And that absolutely proved to be true. But I remember sitting down with FDA, with for example, Gilead, and the initial response was, no, you have to develop them in series, and they came around doing it in parallel. They never asked for, in the end, they never asked for histological endpoints, although Hep C was a liver disease with histological consequences, the same as NASH at some level. So I think one of the nice things about Nash Tag is ideas can come from it that will influence the way people think at agency and the way they think about it at a corporate level and an academic level as well.
If you had a guess, what are the ideas that will come out of this year, or at least the topics on which ideas will come out of this year that will make people pay attention and do things differently?
Well, so this year, I think the major themes are, firstly, the endpoints. The endpoints stink. Okay, and then what is referred to as a gold standard, in reality, is probably the Achilles heel of this disease. It's been a handicap. Histological assessment has been a handicap to drug development. It's just very difficult. You mentioned the kappa. That's one of the elements that makes it difficult. It's an invasive, expensive procedure. All these different things are key. And even the very definition that we've been handed from the NASH CRN may or may not be the most relevant. We have all these components that have variable predictivity for the outcomes that the FDA is most interested in. We have to revisit that. The other thing's going to come out is combination therapies. We have some weak hitters. Generally speaking, many of the agents have not been the sofosbuvir or the penicillin of their field. Finally, I think the mitigation of the placebo effect. We have similarly designed drug studies where you have a 32% placebo response rate per fibrosis stage improvement. It's amazing. We have really well-conducted similar phase 3 studies with 10, 11, 12% rates. That delta introduces so much uncertainty at $500 million to $1 billion phase 3 study. It's very hard to live with. So I think taming and understanding the placebo response, it's not even a placebo response, it's a standard of care response, I believe. So this is going to be another major thing that we consider during this Nash Tag meeting.
That's interesting because I've also heard someone question whether it's a pathologist. I mean, one of the things that drops in the middle of all this, right, is the seladelpar experience. I think it's going to really be a great week.
I'm sure of it.
If somebody bought the virtual package because they couldn't get there for whatever reason, one of the things we all know is that when you go to a meeting virtually, you're far more prone to interruption. So what are the two moments in this meeting when if I'm watching virtually and someone knocks on the door, I throw a shoe at them so that they go away and let me keep listening?
I'm going to say the first session is key from the year in review and the sessions that come after that. And then I would say the second morning. So the first morning and the second morning. And that's not to say that it's not going to be very valuable to do the 2 afternoon sessions, but those 2 mornings, I think, are unique.
First of all, for those who are listening, should know that Michael will actually be joining us for the podcast where we discuss the second morning. So I'm very, very excited about that. It's easy to understand what makes the first morning unique, right? I mean, there's your talk and then the presentation of the 5 major drugs in development. What makes the second morning unique? What specifically about that?
So the second morning, we're discussing evolving therapies and evolving analyses, ways to look at outcomes in studies as well. These are the 2 burning issues, I think, in NASH therapeutics. And I think the insights that we'll get, so if, for example, talking about GLP-1s, it will be Philip Newsom, who's the first author of the New England Journal paper. When we're talking about artificial intelligence, we have Andrew Beck, the progenitor of Path AI, for example. So it's like I'm being able to ask Moses what was the meaning of and what is written on the stone tablets.
I like it. Any other advice to people who will be attending virtually?
Participate. Listening, great, and we certainly have an educational goal, but I think the core of Nash Tag is a vigorous exchange of ideas. So if you have a question, if you have some heretical perspective, send it in. We'll be looking through and get to as many as we can, but speak up and agitate. The field needs to move forward. It needs change, and it needs participation from as many good minds as are willing to participate.
Well, that's great. And we've actually set up these sessions that we're doing to be very different than what we've done at EASL or AASLD or we'll do at any other meeting. At the other meetings, we identify a handful of papers that we think are interesting, and we review them and we make, an hour out of reviewing 4 papers. Here, we're going to quickly summarize the whole morning and let whoever is in the podcast session dive in and try to make sense out of it and bring some of the debate and some of the ferment and the willingness to be bold. See if we can bring that into a discussion that anybody who's attended the virtual meeting can tune in on and make sense out of. We're also going to invite people to ask questions that we then will share either in subsequent sessions or when we do the wrap up on Monday. That is the culture in Nash Tag, right? So we will replicate that as best we can. As we were saying beforehand, I'm going to school right now on 20-year-old kids to figure out better ways to cover hybrid meetings, and it'll be exciting going forward. So anything else you want to say to people who are listening to this, who will be the people who will be attending, I think?
No, I'd like to say that we're excited about this partnership with Surfing the NASH Tsunami. I think any new piece of information or event needs a lens to bring things into focus. And I think that Surfing the NASH Tsunami will provide essentially a real-time lens through which listeners and participants can hear and view the information that's being presented. So I'm hoping this will be a really important adjunct and partnership for Nash Tag. And the final thing I will say, I spent a good part of this year trying to study how to pitch to Babe Ruth. I think I figured it out. And I'm going to present that in the first 30 minutes. So don't miss that.
Okay, what that means is nobody sleep in. Although, if you're not in Utah, the timing is not that painful.
No, that's true.
However, on Saturday morning, we are starting our recording at 6 a.m. Utah time, so that we can get it all done before the event starts. We were planning to do that also on Sunday, spoiler alert, but then Naeem Al-Khoury, who'll be one of the people who'll be with us Sunday morning, brought up the idea that it's daylight savings Saturday night. So it wouldn't be 6 a.m. in Utah, it would be 6 a.m. daylight savings time, which would be 5 a.m. body clock. Since there are sessions on Sunday morning to constrain us, we're going to start an hour later. We're going to start as if the clock didn't change. An early start nonetheless.
Where are you based, Roger?
I'm in Pennsylvania.
Okay. So you'll be suffering more than us. Oh, no, a bit less.
Less. No, it's easier for me. It'll be 8 in the morning and 9 in the morning for me.
We'll make sure the speakers and and anyone working with you on Surfing the Nash Tsunami is well supplied with coffee during the...
Coffee will be really important. So thank you for that. So Michael, I'm going to let you go. You go to your dinner. I'm going to wrap this up, and I will see you on screen Saturday afternoon. And I don't know if you know this. I have a daughter and a grandson in Chicago that I've not gotten to see for a long time, but because of the pandemic and because between her job and his daycare, there's never been a situation where anybody could afford to have to quarantine. But I'm exactly one vaccine away from being able to come to Chicago, which I'm tremendously excited about.
Well, congratulations. It does feel like not just that it's the literal spring, I do feel like the thaw in this pandemic is well underway as well. So as we mentioned before we started speaking this evening, Nash Tag was the last conference many of us went to before the pandemic really took on its momentum, and it's the first one many of us have gone to since the pandemic began. So hopefully the same momentum will carry you to seeing your granddaughter.
Well, and everything else that we've got to do. Hey, Michael, thank you so much. See you on the podcast Saturday, and have a safe flight out tomorrow, okay? And enjoy the skiing.
Thank you, Roger.
Bye-bye now.
For our last section, Roger Green discusses how SurfingNASH plans to cover Nash Tag.
And now that you've heard from Michael and you've heard from Vlad, I think they started off with exactly the same story about HepDart and skiing. Let me take a minute and talk about our coverage of Nash Tag. As I just discussed on our conversation with Michael, our goal is going to be not to merely present papers, but to help people, particularly those who are attending virtually, but anybody who wants to listen, catch some of the feeling and some of the flavor of the ferment and the intellectual development and insight development that takes place over the course of those 2 days. So what we will do is we will record one episode for each session. Now, because they're in Utah, the sessions run from 8 in the morning until 12 Utah time, and then again from 4 in the afternoon until 8 Utah time. Now, 8 in Utah is 10 in New York, 3 in the morning in London, 4 in the morning in Paris, and literally the next morning in India, for example. So what we've set out to do, we wanted a blend of European and American key opinion leaders and commentators. So the first thing that's exciting is Donna Cryer, who loves this meeting as much as we all do and cannot be there, will be with us all 5 sessions, the 4 conference sessions, and then the Monday night wrap-up. Louise would like to also, but she has to go spend Sunday giving vaccinations as part of the work she's doing with NHS right now on the COVID vaccine program in Europe. So she'll be with us for Friday, the 2 on Saturday, and again on Monday night. I will be there for all 5. Beyond that, we have really, I think, a great assortment of people coming to join us on the Friday session, which we will be recording starting an hour after it ends, so at 1 o'clock in Utah, at various times in different places, and intend to drop about 8 o'clock in Utah, which would be 9 in the Midwestern U.S., 10 in the Western U.S., and while you're sleeping in Europe the next morning in India. We will have Manal Abdul Malik, a renowned key opinion leader and great friend of the podcast who's been with us a bunch, and also Ian Rowe, who, if you've listened to Ian on the podcast, I think takes fascinating and really different ways of looking at things. This will be mostly the discussion Michael talked. It'll be about Michael's talk about the last year and pitching to Babe Ruth, which would be fascinating for Americans at least. And then 5 different drugs they'll be looking at. You can find the drugs actually if you look at the Nash Tag program. So that will drop late Friday. Now, we don't want to start taping right after the Friday session because it'll be impossible to do in Europe, frankly. So we're getting up very early Saturday morning, 6 a.m. Utah time, and we'll be recording then, and that session should drop sometime between when the Saturday morning Nash Tag session ends, for those who are virtually attending, and when the Saturday evening session begins. With us Saturday morning, we'll have Stephen Harrison, and we'll have Jörn Schattenberg from Germany and Naeem Al-Khoury, both good friends and frequent participants on the podcast. Saturday's morning session, the one that Michael's excited about, will end at noon. We will come up at 1 o'clock and drop Saturday night on that. Michael will be one of our guests for that session, and the other one will be Ian Rowe again. And then on we will record on Saturday night, and we'll come back on Sunday, and our key opinion leaders for that will be Jörn Schattenberg and Naeem Al-Khoury. The normal Monday night session, Vlad Ratziu will be joining us. We are nailing down who the second key opinion leader will be now. I'll be able to announce that by the first podcast. And join us for the podcast. We will have an open way for you to get us questions during the course of when you're hearing the recordings. We will not be able to answer the questions on the recordings, but we will collate them and bring them back to the subsequent episodes and then to the Monday night roundup. So if you want the full Nash Tag experience and you can't get to Park City, then except for the fact that you can't ski, in the scenario I'm pointing at, go to the sessions, and then between the sessions, come to the podcasts, listen, send us questions, engage us, and I think it'll be a great experience for everybody. We're really excited about this, and we're thrilled to be part of it, and frankly, thrilled by Michael's kind words that I did not expect, and we're gratified to hear. So with that, I'm going to sign off. I want to thank Mike Wilson, who will be pulling this tape together, and Eric Rounds for everything he does. There won't be conversation, so I can thank Polly for being Polly, but not for anything that he's doing this week. I want to thank Vlad for making time this morning, Michael for making time at dinner, and all of you, our listeners, who make all this worth doing. I'll take one more minute, and I will tell you that 3 of the 4 biggest weeks this podcast has ever have been the last 4 weeks. And we are now at a much higher volume than, frankly, I thought we'd be for another year. So thank you for coming and listening and paying attention and downloading episodes and all that good stuff. We will see you on the podcast, and I hope we will see you this weekend at Nash Tag. Trust me, it'll be worth it. Stay safe, surf on. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.